Okay, here's our video on the notes for laboratory quiz 3. This is our quick little guide to lab 3. So let's go through the different laboratories. Laboratory 3 will cover labs 19, 21, 22, 23, and 24. As a quick review, 19 was the glucose tolerance test. 21 is on pressures and the startling forces. 22 was on red blood cells, including the count, the hematocrit, the hemoglobin, MCV, MCHC, and blood typing. 23 is on white cells, total count, differential count, and 24 is on the ECG and then the ECG and pulse. So those are the laboratories that are on it. It will be taken online. And we'll deal with the other information on the announcement that you can look at. The information below is certainly not a complete list, as always, but it does give you a few areas that you should concentrate your efforts on. As usual, anything in Fox or the supplement is fair game if it's indicated. And I did pull one question from the back of the Fox laboratories for this quiz to reward those people who are doing what they're supposed to. So let's kind of look at each quiz a little bit, each lab a little bit. So lab 19, remember that's the supplement only. There's no Fox correlate. So just look at the information in the supplement. That's where we did the glucose tolerance test. So know how we did it. Ate a whole bunch of jelly beans, measured glucose initially before eating the beans, and then every half hour after that. You know where the normal ranges for glucose and the values, what the tests look like, what's an abnormal test. So if the range is 70 to 100, what happens if you're under 70? What happens if you're over 100? What's the level to become diabetic, as an example? What are the expectations for blood glucose and urine glucose over time for people? So what's a normal test versus an abnormal test? You should know how blood glucose is controlled, so the relative contributions of insulin and glucagon. You should know the terminology. So there's a little section, a couple questions at the back of lab 19 in the supplement that ask about the different things associated with it, and the disease and things like that, and then the different types of diabetes mellitus. So especially type 1 versus type 2 versus gestational diabetes. So you should go back and look at those. In the supplement, there's rat hormones, okay, and the rat hormones are omitted from this quiz. So we'll just cross those out because we're not going to look at rat hormones from that. So lab 21. Lab 21 is in the supplement, and it's also in Fox 7.6. There are actually a couple things in the supplement, and one major thing in Fox 7.6. And then the rest will be omitted, as we'll see when we get down to lower. So Fox 7.6 is kind of the big portion of this. It's the measurement of mean arterial blood pressure. So you should know the values, what normal ranges are, things like that. You should know the difference between hypertension and hypotension and the terminology associated with those things. You should know the calculations of pulse pressure and mean arterial pressure. So it's lab. I would have to give you those formulas. Remember, pulse pressure is the difference between systolic and diastolic pressure, while the mean arterial pressure is calculated as 1/3 systolic plus 2/3 diastolic pressures, or 1/3 pulse pressure plus diastolic. You should know the effective body position on blood pressure. So in the positions we tested, which would have the highest blood pressure, which would have the lowest. You should know about how to measure blood pressure in terms of pumping up the cuff and what we hear and when we hear it and what we note. And then you should know the changes in cuff pressure as results to systolic and diastolic sounds. So why do we hear the first sound, and why do we attribute to that systolic pressure, and why do we hear the last sound and diastolic, and why don't we hear it above systolic, and why don't we hear it below diastolic, would be all things to consider. You should know how to measure venous pressure. That was in the supplement. You should know the values, the normal values for venous pressure, so like 4 to 10 millimeters of mercury. You should know how to perform the test with the hand lifting up and the vein collapsing and measuring the height above the heart. That difference divided by 13.6 to get the put it in millimeters of mercury units, and that's the calculation. In addition, we didn't really talk about it, and it's mentioned. There is some questions on the Starling forces, right? That's in the supplement only, and know how to calculate net filtration pressure. Learn it now because we're going to see it again when we get to the kidney. So the more you learn now, the easier it'll be when we get there in lecture. Know what happens at each end of the capillary, so the arterial end versus the venous end, and which favors filtration and which favors reabsorption, right? So that's what that's asking. Know how to calculate the net filtration pressure and be able to interpret the findings. Again, it's lab. I would have to give you the formula. In terms of the other things you might think you're responsible for, right? The goldfish tail is cool, but we've omitted it from the quiz, so don't worry about that. The human heart parameters we didn't talk about, so that's omitted. The heart sounds in 7.5a, that's omitted. And the fitness test in 7.7 is also omitted from the quiz. So those 3 points are the major points of Lab 21 that you would have to know. So continuing on, we'll look at Lab 22. Lab 22 is a significant lab, probably the most significantly represented, because we did two entire Fox experiments in Lab 22. The first one was Fox 6.1, so make sure you know the information in there and look at the information at the back. Also, since we're talking about Fox, 6.2 and 6.3, which are the white cells and the blood typing. So you should understand the factors associated with red blood cells. Should know the normal values for total red blood cell count, hematocrit, hemoglobin, the MCV, the MCHC, right? So all those things, you should know how to calculate these when appropriate. So given values for total red blood cell count, hematocrit, and hemoglobin, how do you calculate MCV and MCHC? And then how do you figure out what those things mean? Know the problems associated with the abnormal amounts of these factors. So primarily talking about anemia and what occurs there. You should know the terminology and the definitions as well. So that's Fox 6.1. In Fox 6.3, that's the blood typing. So you should know what the blood types are and how they're antigens on the red blood cell surface, so just glycoproteins. Know the difference between the ABO group and the RH group. Know who can get what blood type for transfusion. Understand how we determine blood types in the laboratory with the agglutination and what happens there. And understand also the hereditary aspects of the blood type, so the ABO grouping and the RH grouping for that. For Lab 23, that's Fox 6.2, that's the white cells. The white cells, we did two things. We did a total white blood cell count and a differential white blood cell count. So the total white cell count, you should know the numbers and how we did it with the hemocytometer. For the differential, you should know about staining the slide and counting the different types of white blood cells. You should understand the normal values for each. You should understand the associated abnormal count. So what's leukopenia versus leukocytosis and what's associated with those. You should understand the other terminology and definitions associated with these. What's not on here, which could probably also put, is understand the percentages of the individual white blood cells. That's from the white blood cell count. And understand the morphological characteristics. What do the white blood cells look like? I'm not going to ask you what's their function. So as an example, on the lecture exam, I would not feel bad asking, which white blood cell works well against parasites, right? And that would be eosinophil. I'm not going to ask you about that functional stuff in lab, But I would ask you, hey, what makes up 2 to 4% of the white cells, which would also be eosinophils or something like that. So that's lab 23. Lab 24 is from the supplement in terms of the Biopack Lesson 5 and Biopack Lesson 8. In Lesson 5, we looked at the ECG, so you should know the basic ECG waveforms and how they correspond to heart activity. So where's ventricular depolarization, repolarization, and so on. You should understand the QT and TQ intervals. The QT is systole, the TQ is diastole. Understand how those change with exercise. You should know changes in the respiratory cycle with breathing, so the difference between inhalation and exhalation. You should understand terminology and definitions as usual. And then for lesson 7, that was the ECG and pulse, so you should understand about the pulse plethysmograph and how we measure the pressure wave. You should understand the relationship between the ECG and the pressure wave, that the ECG, right, that electrical recording precedes the physical event of the heart contracting. The heart contracting pushes the blood out, and that creates the pressure wave or the pulse wave. That's what we feel when we palpate the pulse. You should understand the effect of moving the hand height. So it should get bigger because of gravity when the hand is below the heart, and it should get smaller because of gravity when it's above the heart. The ECG doesn't change in size because it's electrical, and it's the pulse pressure wave that changes size. You know how we measured them and how they're related. So that's just a quick little summary of the different things you should pay attention to for this next lab quiz.